Hello, class. Hope everybody is ready to go and start on chapters 6 and 7 today. We are going to be talking about cell division, different types of cell division. So, we've learned about cells, but how do they make new cells, right? That's part of the cell theory, is that all cells come from other cells. So we are going to get into that right now and talk about mitosis and meiosis. So let me open up these files for you. Here we go, chapters 6 and 7, cell division and inheritance. So stuff we're going to talk about, different types of cell division, and then go into two specific types. So this is going to be just prokaryotes versus eukaryotes for the different types. And then mitosis versus meiosis. So these are different cell divisions that occur in your body, and how sex is determined, and then what happens if there are problems during cell division and there's the wrong number of chromosomes in a cell. So in this lecture, I think we'll just get through types of cell division and mitosis, and then we'll cover the rest in a little bit. Okay, so let's just get through a little bit of terminology. So as we're talking in this lecture and going forward through the rest of the semester, everybody knows what all these terms mean. So a cell's complete set of DNA is its genome. You may have heard of genome sequencing. That means sequencing all of the DNA in your cell. So every cell should have the same genome. So it's, you just need one cell to get that, to look at the genome. We have our DNA organized into chromosomes. Humans have 23 pairs or 46 chromosomes total. So our genome would be all 46 chromosomes, okay? And the different genes in every single one of those chromosomes. So one chromosome is just a long strand of DNA, and they have pairs, okay? When they're replicating, right, when a cell replicates, that genome needs to replicate too, so that each new cell, right, the two cells that you get after it splits, each has the full genome in it. So when they've condensed and they're replicated, they actually end up looking like this. There are these two pieces that are just kind of held together right there. Each of these pieces, there's this one and this one, those are called chromatids. So a chromatid is just a chromosome that's replicated, and it's one, it's one piece of that. They're like identical twins there. The prokaryotes, right? So organisms that don't have a nucleus, and their DNA, as we talked about way back in chapter 3, I think, they don't have their DNA linearly like that. So this is a double helix, right? They have their DNA in one circular strand like this. So it's not going to condense into chromatids the same way that it does in eukaryotes. So prokaryote cell division is a little different, and that's the first thing we're going to cover, and then we'll talk, most of the rest of this chapter is going to be about eukaryotes. So we'll start with prokaryotes. When they divide, and remember, prokaryotes are all single-celled organisms. It's one cell, and it divides, and then you have two cells. This is what, when bacteria are replicating, you start with one, then you have two, then you have four, then you have 8, right? They just keep replicating. And every single one is identical to the one that it came from. So you start with 1, and it replicates into 2, and these 2 at the bottom that I'm coloring in blue are identical to the one that started, that I'm coloring in green here, okay? They're genetically identical. When they divide, that's called binary fission. So bi means 2, and fission means to divide. Fusion is coming together, fission is coming apart, okay? So we've divided into 2 parts. The way that this fission occurs is first the DNA is replicated. You need 2 copies of your DNA so that each cell gets one, each new cell gets one, okay? So they basically unzip. And let me draw a little picture for you of that. Okay, so we've got here this strand of DNA. So this is our prokaryotic circular DNA. It's inside a bigger prokaryotic cell, okay? The first thing that needs to happen is this DNA unzips. So instead of being attached like that, it's going to come apart from itself. So now we've got two strands, but they're not twisted up together. Each of these is then going to attach to a side of the cell membrane of this prokaryote. And then as the cell membrane pinches off, we've got this piece of DNA attached over here, got this piece of DNA was attached over here. The cell membrane can kind of pinch off and make two new cells, okay? And each of these is, this will, as these separate, they will then replicate themselves. Okay, so they separate and replicate. So each of these gets a full double-stranded piece of DNA as well. Okay, so that is binary fission. In prokaryotes. So let's go back to that slide that kind of just walks it through. Okay, so our first step here was replicating that DNA. It unzips, and then each of those unzipped pieces of DNA, right, it unzipped, each of those gets replicated. So now we've got two circular pieces of DNA instead of one. Each of them attach at a different spot on the main cell, and then the cell pinches off and forms two cells. Okay, so now we have two identical daughter cells. That is cell division in prokaryotes. And that's all we're going to say about them. We're moving on to eukaryotes now. Eukaryotes, it's a little more complicated because it's not just one piece of DNA, right? We've got 46 pieces of DNA, right? 23 chromosomes, each one paired up. This is chromosome 1, chromosome 2, chromosome 3, right? We've got 46 of these. It's much more complicated in a eukaryote just because there are more pieces involved. And there are organelles and things like that involved also. And a nucleus, right? We've got to split the nucleus. So eukaryotic cells have different phases. There's this interphase, which is the growth phase of cells. So this is when they're growing and DNA is dividing, getting ready for mitosis, if it's going to happen. And also just doing its usual cell stuff, right? Doing cell stuff, right? Making proteins, things like that, right? Our cells have a purpose. If it's a neuron, it's sending messages, right? If it's a blood cell, it's, well, our blood cells divide in the bone marrow, but it would be, you know, getting oxygen, or if it's a white blood cell, fighting disease, right? They're doing things other than just dividing all the time. That's happening in interphase. And then we get into mitosis. During mitosis, this is when the nucleus is dividing. So this is the DNA is getting divided from the nucleus. And then also everything in that cytoplasm, all of the mitochondria, and if they're chloroplasts and ribosomes and endoplasmic reticula, right? All that needs to get divvied up so that each daughter cell, right? You sort of go from one parent to two daughters. Each of those gets all the stuff that's inside too. So if we look at a picture of this cell cycle, most of this, all this gray going around, this is during interphase. This is cells doing their cell stuff, okay? They're growing, they may be replicating DNA, right? They're growing again, they're doing all of that. Mitosis is just a small part of that. Different cells, though, have a different cell cycle, different cell type. So every cell is not on the same schedule. So as I've got written down here, some cells are going to move through this quickly, and some may never divide. So if you think of your skin cells, right, if you rub your skin, you can see cells coming off, right? especially if you have dry skin, you can really see that. They're constantly dying, and your body is making new skin cells all the time. But some cells, like your heart or your brain, like neurons, they might never divide even once in your lifetime, which is why some things like a brain injury are so hard to recover from. If those neurons are damaged, you're not making new ones. So different cells, if we look at this cycle, some cells may spend all of their time in interphase, right? But some cells, like a skin cell, might spend most of its time mitosis, like I'm going to divide, I'm going to grow and replicate, and then I'm going to divide again. They're just constantly dividing. So different cells have a different cell cycle, just their timing of things. Whether they're dividing a lot or not is determined by this control system. So there is this internal control system that says like, okay, you get to divide now or no, right? Just grow and do your stuff and don't divide. So there are these different checkpoints and controls that will make sure cells are on the right cycle. The first checkpoint, and you guys don't need to memorize each checkpoint, but this is just to kind of understand how cells know and why, if you think of the human body with all of our millions of cells, most of the time things work well. There's a checkpoint that makes sure there's enough nutrients available for replication, right? This is going to take energy. We need to have enough extra of the components of DNA to replicate the DNA, right? DNA is made up of A, T, G, C. If you're going to make 2 copies of it, you got to get more A, T, G, and C. Make sure you've got enough of those, right? Make sure that DNA looks good. You don't want to make a copy of bad-looking DNA. Make sure it's not damaged, right? So there are these checks, like, okay, we've got our nutrients, everything looks good, you can go. After the DNA is replicated, there's another checkpoint, okay? Did that replicate okay, or is that damaged DNA? If that DNA is damaged, you don't want a whole new cell filled with damaged DNA, and that cell would then replicate, and all the DNA that comes out of that would be damaged, right? So if stuff is bad, it's not going to continue. And then there's other checkpoints to make sure during mitosis, things are lined up properly and everything is going as it should. Okay, and then we'll get into, there are some enzymes that if things don't look good at these checkpoints, they'll stop it, and sometimes they'll even just program the cell to die. Okay, you look bad, go away, you're going to cause problems for this whole organism. So let's talk now in a little more detail about mitosis. In order for cells to replicate, we need 2 copies of the DNA, right? And we've talked a tiny bit, but this is a little bit more about how that DNA structure makes replication, perfect replication is possible, right? We can actually attain perfection because there's this complementary base structure, which means that every A pairs with a T, every C pairs with a G, right? If you have an A, you know what the one on the other side is going to be. If you have a G, you know the other one is going to be a C, right? That means if you split them apart like they've done in this second strand here, you can fill it in perfectly, right? So we split these DNA apart, these 2 strands, and they have filled in with the complementary bases in orange here. And now we have 2 strands at the end, and they're identical. These 2 daughter strands are identical each to the strand that we started with. Each is half of the old strand and half new, okay? Because of that, because there's this paired structure, we can split them apart, and we know that each cell is going to get half of an old, half of a new, but their DNA is going to be identical. Okay, so the different parts of mitosis, these are our phases. Interphase, which we already talked about. That's during the cell growth and replication. And then there are 4 other phases: prophase, metaphase, anaphase, telophase. So you're going to need to remember this order. It goes I, P, M, A, T. And you can come up with a mnemonic device like I'll probably make apple tarts, right? So during interphase, the chromosomes are replicating, checking, making sure they've got enough nutrients, good to go. Prophase, okay, we've got our replicated chromosomes. And after they've replicated, each one is a double strand, okay? and they've replicated, but they stick together, okay? These are now like our twins that are, they're identical and they're stuck together. They're going to condense, just get a little shorter. If you think of like if you have a long strand of toilet paper, if you fold it up, it's going to be easier to move it from one side to another than if you've got, you know, the whole roll of toilet paper unrolled, right? So they get condensed, all squished together, make it easier to divide. And our nucleus is going to break down. You can't divide your DNA, you know, half chromosomes go on one side, half go on the other if they're stuck in the middle of a nucleus, right? So that nucleus is going to disappear, getting ready to split up the DNA and split the cell. Okay, metaphase, everything goes to the middle of the cell. And I'm going to show you pictures, but metaphase, everything lines up in the middle. Anaphase, they get pulled apart. And telophase, a new nucleus is going to form around each of those, all of those chromosomes, some on one side, some on the other side, and the cytoplasm is going to divide also. That's cytokinesis, cell division here. Okay, so let's look. Here we're at interphase, the top left here. Cell's getting ready, okay? And then we're moving kind of clockwise around here. Prophase, the chromatin is condensing. Now you can see the DNA here, right? We can see all that DNA. When it's all unfurled, it's so tiny, but you can actually see condensed DNA in our microscopes. So you go from these little squiggles up here that you can barely see to thick pieces of DNA in prophase. And this nucleus is breaking down, and you can start to see these little spidery web things here. That's the spindle that's going to pull DNA to one side and the other side. It forms on each side of this cell. Okay, metaphase, all of the chromosomes are lined up in the middle. Nice, right down that like equator there. And then anaphase, they get pulled apart. So you can see we had started with our chromosomes were attached. And here you can see they're getting pulled apart. So one piece is getting pulled to this side and one piece to this side, right? Pulled to the top and bottom. Still anaphase, they're getting pulled apart. But here you can see it's starting to pinch a little bit, right? It's starting to divide the cell, which would be cytokinesis. And here we're at telophase, and we can see, look, new nuclear membrane has formed. So we now have a new nucleus, and we can't see the DNA as well anymore. It's spread out. It's not condensed. Okay, this is just looking at each phase individually. Prophase, our nucleus is breaking down. You can see it's got this dotted line going around it now. Our replicated chromatids, you can tell they're replicated because they look like X's instead of just a line, okay? You can say our replicated chromatids. They're condensed so we can see them, and the spindles are forming. We can see our little spindles are starting to form up here, and they grow longer. There are these fibers that are going to pull the chromosomes. Okay, so that's prophase. Next is metaphase. These chromatids are lined up in the middle, right? And so when the spindle fiber pulls, it's going to pull one to one side, one replicated piece to one side, the other replicated piece to the other. So that's metaphases in the middle. Anaphase, they get pulled apart. And telophase and cytokinesis usually happening at the same time. So the telophase, anything that ends in the word phase is talking about the nucleus. So what's going on with the DNA and the nucleus? Okay, the chromosomes are uncoiling. We can't see them as well anymore. This nucleus is reforming. You can see that nuclear membrane reforming. And the cytokinesis is splitting everything else up, right? This cell starts to divide into two. Okay, we're going to stop here. I think that's enough for now. And then we will kind of review mitosis and get into meiosis in the next chapter. Bye-bye.